We tend to think of the chloroplast as a uniform, standardized component of plant biology. However, its physical structure is highly variable, changing according to the specific organism that houses it. Lower plants, for instance, lack a standard template. They utilize highly specialized morphologies that differ widely across species. This schematic illustrates variations found in algae. On the left is a cup-shaped structure native to Chlamydomonas. On the right is a helical, spiral-shaped chloroplast characteristic of Spirogyra. The variation continues. Eulothrix relies on a horizontal girdle shape, while Zydenema contains a multi-pointed star structure. These specialized anomalies stand in contrast to higher plants, which have moved away from these varied shapes in favor of a uniform, oval-shaped chloroplast. While lower plants utilize varied morphologies to suit specific evolutionary niches, higher plants have converged on this oval architecture as the standard template for energy production. To understand the efficiency of this standard oval chloroplast, we have to examine its outer boundaries. When we slice through the organelle, we reveal a distinct secondary layer underneath the outer shell. The chloroplast is contained by this double membrane structure. This boundary isolates a space that operates with unique independence, defining the chloroplast as a semi-autonomous cell organelle. This semi-autonomous nature comes from containing its own genetic instructions. Floating within the internal space is a looping, double-stranded circular DNA structure. It also possesses translational machinery. These surrounding clusters are 70S ribosomes, allowing the organelle to synthesize its own proteins. By possessing independent circular DNA and 70S ribosomes, the chloroplast effectively functions as a self-contained engine, operating with independence from the host cell's central nucleus. We can now move inward, past the double membrane, into the internal volume of the organelle. The interior is filled with an aqueous matrix known as the stroma. In this structural map, the fluid space, color-coded blue, is the stroma, the exclusive site of the dark reactions. Suspended within this fluid are physical structures, groups of flattened, disc-like thylakoids stacked together. These stacks are called grana. The grana serve a separate function from the stroma, acting as the exclusive site for the light reactions. These stacked discs do not operate in isolation. They are physically connected to one another by structural bridges called intergrana. This spatial compartmentalization prevents the light and dark chemical reactions from interfering with one another, ensuring a precise division of labor within the organelle. To find where light is actually captured, we need to focus on a single thylakoid disc within one of the larger grana stacks. The surface of every individual thylakoid is not smooth, but is studded with microscopic nodes. This macro view of a thylakoid surface reveals a uniform grid of raised nodes. These are quantisomes. The quantosome is the absolute smallest functional unit of photosynthesis. It earns this classification because it serves as the physical housing for the chloroplast pigments that trap light. A plant's ability to harvest energy relies on a massive, distributed array of these localized units, rather than a single large receptor. Looking directly inside a single quantosome reveals an interior composed of a specific, unchanging number of pigment molecules. This array illustrates the exact count. One quantosome contains exactly 230 chlorophyll pigment molecules. Breaking down that total, the array contains 160 molecules of chlorophyll A. The remainder of the unit consists of exactly 70 molecules of chlorophyll B. We can isolate one single chlorophyll pigment molecule from this cluster to examine its physical anatomy. Every chlorophyll pigment is divided into two distinct structural parts, a wide geometric head and a long linear tail. The head measures exactly 15 by 15 angstroms, while the tail extends 20 angstroms in length. The tail is an explicitly hydrophobic phytol chain and contains exactly one double bond. These rigid, predictable dimensions allow the pigment molecules to pack together into the 230-molecule array, creating a dense, continuous surface area for photon absorption. If we focus exclusively on the 15 by 15 angstrom head of the molecule, the mechanism of absorption becomes clear. This square head is a magnesium porphyrin complex. As shown in this layout, the center of the complex holds a single magnesium ion. This central magnesium is anchored by four surrounding structures known as pyrrole rings. A single pyrrole ring has a specific atomic composition. It contains exactly 1 nitrogen atom and 4 carbon atoms. The hydrophobic tail we examined earlier attaches to this head at a specific anchor point, the 4th pyrrole ring. This magnesium porphyrin head acts as a highly ordered geometric antenna, structured at the atomic level to trap the resonance energy of light. The difference between chlorophyll A and chlorophyll B comes down to a single location, the 3rd carbon located on the 2nd pyrrole ring. In chlorophyll A, this specific location holds a methyl group. This configuration yields the exact chemical formula for chlorophyll A, C55H72O5 N4Mg. In chlorophyll B, that single methyl group is swapped out for an aldehyde group. This swap yields the formula for chlorophyll B, C55H70O6 N4Mg. This minute atomic difference alters the pigment's absorption spectrum, allowing the plant to capture a slightly different wavelength of light. Following this zoom, we move from a single bond up to the 230-unit quantosome, out through the grana, and back to the double membrane organelle. Photosynthesis is the result of this hierarchical physical engineering, where the structure of every scale, from the organelle down to the specific aldehyde group on a porphyrin ring, contributes to the capture of energy.